So, let's start with asking the question why one would do a BLAST search. The hypothesis underlying sequence similarity searching, also referred to as homology searching, is that if sequences are similar at some statistically significant level, they share a common ancestor. If they are similar, then they are likely to have a similar structure. And if they have a similar structure, they are likely to have a similar function. As such, a sequence similarity search often provides the first information about a new DNA or protein sequence. It allows you to infer the function of a sequence from similar sequences. Most proteins are modular in nature, with one or more functional domains occurring within a protein. The same domains may also occur in otherwise dissimilar sequences. The BLAST algorithm enables to locate and align domains in a protein sequence. You can establish phylogeny using BLAST. You can map DNA from an unknown location. The local alignment approach also means that a messenger RNA can be aligned with a piece of genomic DNA, as is frequently required in genome assembly and analysis. If instead BLAST started out by attempting to align two sequences over their entire lengths, known as a global alignment, which we covered in the previous session, fewer similarities would be detected, especially with respect to domains and motifs. So, let's start covering the principles of BLAST. BLAST and its descendants are probably the most used database search programs. BLAST stands for Basic Local Alignment Search Tool and was introduced by Altschul and colleagues at the NCBI in 1990. From the previous sessions, we have seen that the amount of data in the sequence databases is growing exponentially. We can also readily see that the rigorous pairwise alignment methods already described are too demanding in memory and computation time for routine searching of large databases. The time to compute an alignment between a string of length n and a string of length m is seen to be proportional to n times m. The need for something faster gave rise to BLAST and other such programs. BLAST compares nucleotide or protein sequences to sequences in a database and calculates the statistical significance of matches. Using a heuristic method, BLAST finds similar sequences not by comparing either sequence in its entirety, but rather by locating short matches between the two sequences, working on the assumption that good alignments contain short lengths of exact matches. So, BLAST can be divided into three parts. First is the setup stage, where the BLAST input is given and the parameters are set. The second part involves searching for local alignments between a query sequence and a sequence in a database. And the third part is where p-values are produced for local alignments and the alignments are ranked based on those p-values. The output of BLAST is a list of high-scoring local alignments together with a measure of the probability that such matches would occur by chance. So, the first part. In the setup part of BLAST, the program reads in the query, the search parameters, and the database required to carry out the search. These are most often, inputs that the user gives. And if you have selected filters, then the program will read these in too. And we will cover low complexity and other repeat filters further along. So here is an example of the setup step. I'm sure most of you are familiar with this page. This is the web interface on the NCBI website for submitting a BLAST-P search. The link to this page is given on the right. On this page, you would give the program your sequence of interest by pasting it in this box or uploading it by clicking the Choose File button here. You'd specify the database you want to search your query against. You can even narrow your search down to a specific genus or a species by entering the genus or the organism's name here in this box. And then you select the BLAST protocol. I'll explain the differences between all these different types of protocols later. You can adjust the stringency of your search by giving a lower e-value cutoff, and I'll explain more what the e-values mean and how they're calculated in a little while. You can turn the low complexity filter on or off. Again, I'll explain what that does in a later slide. And finally, once you're happy with the input and the parameters you've given, you'll submit the search by clicking on BLAST. And the second part, the BLAST parameters that were set are used to search for local alignments. First, the query sequence and the database sequences are broken into words. For a protein-protein search, a word length of 3 is typically used. For a nucleotide-nucleotide search, a word length of 11 is typically used. BLAST then scans the database looking for matches between the words in the query and words found within the database sequences. For nucleotide-to-nucleotide searches, these matches must be exact. For protein-to-protein searches, the score of the match, as determined using a substitution matrix, must exceed a specified threshold. Now let's assume a protein sequence like this one. The sequence will be broken down into 3-letter words, as demonstrated here. And the sequences in the database are also broken down in a similar fashion. The words in the query are then matched with the words in the database as such, and the locations noted. After that, each word match is scored. The score is computed by assigning a value to each aligned pair of letters, and then summing these values over the length of the alignment. For protein sequence alignments, scores for every possible amino acid letter pair are given in a substitution matrix, where likely substitutions have positive values and unlikely substitutions have negative values. The default matrix used by BLAST-P is BLOSSOM62. For nucleotide alignments, BLAST uses a reward of +2 for aligned pairs of identical letters and a penalty of -3 for each non-identical aligned pair. Let's take an example of this protein sequence and score some of the words derived from it. So we use a scoring matrix such as the one shown here on the left to score each of these words. What would be the best score that each of these words could accomplish? Remember, a BLOSSOM substitution matrix is based on observed aligned protein sequence blocks of very conserved regions of protein families. So the scores in the matrix show the frequency at which an amino acid has been observed to be substituted with another amino acid in nature. The best score is along the diagonal line of the scoring matrix. So, for example, the best score for amino acid M will be 5. Here. For R is also 5. And so giving us a total of 15 for the word MRR. In this way, scores for all the words are calculated. The words that are below some particular threshold are then filtered out. You can specify this as a BLAST option. Example, when threshold T is 13, the word AAI is removed from the list. This is just a way of removing uninformative parts of the sequence that aren't going to be very useful when searching through a database, and they're going to occur a lot of times. The words that are retained are then used to seed the next step, which is constructing an ungapped alignment. The words are extended in both directions into gap-free extensions for as long as the cumulative alignment score increases or stays the same. Gap-free extensions with alignment score above a given threshold are used to seed gapped extensions. Gapped extensions, also known as high-scoring pairs, scoring above an empirically determined cutoff score S, are retained. An HSP is the fundamental unit of BLAST algorithm output. A high-scoring pair consists of two sequence fragments of arbitrary but equal length whose alignment is locally maximal and for which the alignment score meets or exceeds a cutoff score S. A set of HSPs is thus defined by two sequences, a scoring system and a cutoff score. This set may be empty if the cutoff score is sufficiently high. After the gapped extension step, we end up with a list of HSPs. Next, BLAST assesses the statistical significance of each HSP score. To determine the probability of the HSP alignment occurring by chance, and in biological terms to determine that two sequences are descended from the same common ancestor. To analyze how high a score is likely to arise by chance, a model of random sequences is needed. In brief, how this is done is that we start with our query sequence and calculate all possible combinations of those amino acids in random. And then we align all those combinations to the database sequence we found and we calculate the scores for each of these alignments. The fraction of random sequences with a score greater than the score the HSP had obtained gives us the likelihood that our alignment was just at random. If that value is very low, it means that our score is significant. If that value is very high, then it means that there is a good chance that our alignment was one of those random ones that we generated. So you can say that a p-value is the probability of finding exactly a high-scoring pair with score greater than or equal to S.